Good morning, good afternoon, and good evening, everyone, and welcome to this Inside Scientific and Scientist.com webinar titled Towards Automated Spike Sorting Using Artificial Intelligence on a Cloud-Based Platform. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. This webinar has been sponsored by Diagnostic Biochips, so a big thank you to them for helping make this event possible. We're very fortunate to be joined by Dr. Fan Wu, VP of Product Development at Diagnostic Biochips, and Lidor Spivak, a PhD student in the Stark Lab and the Sagal School of Neuroscience at Tel Aviv University. And they will be discussing the use of artificial intelligence to automate electrophysiological data analysis and a new cloud-based platform. And with that, I'd like to welcome our first presenter, Fan Wu. Thanks so much for joining us today, Fawn, and the floor is yours whenever you're ready.
Thank you, Andrew, for that kind introduction. My name is Fan. I've been working at Diagnostic Biochips for over 7 years on new product development, and it's my honor to present DBCloud to the public for the first time over this webinar session. So let me share my screen. And so most of you, some of you are customers of DBC. You might have heard of us because of our high-density silicon probes. We are very good at developing these high channel count electrophysiology electrodes to acquire very large, complex data sets that can contain lots of single neurons. But as we realized, there have been a lot of bottlenecks for users to deal with this large and complex data sets. So over the past few years, we've been working on a cloud solution to help solve some of the data challenges. So in this presentation, I'll go over some general challenges in ePhys research field. I'll go over some of the core DBCloud framework and technology that's been built already. And as an example of how we can collaborate with external labs and researchers, here we will introduce a second presenter to show you a new feature involving AI to help automate spike sorting. So we have noticed that in general ePhys community, there's a very big challenge in setting up new lab because the infrastructure can be very expensive to build and takes a lot of time to set up. This can involve hundreds of terabytes in storage and high-end computing resources. And to deal with this data set, typically students would have to write their custom code that is difficult to support and share between different generations of students even, let alone across different electrophysiology labs. And also, typically, when a student acquire a large set of data for their own research, that data set is only being consumed by that student only. So we see a potential in somehow utilizing the same data set for other research across the field. So to bring solutions to according to those challenges we have already identified, we propose to bring ePhys to cloud. And this allows users to do all the data storage and computation all from a simple access with a browser. And you can just subscribe to the service and have access to all the services that that's offered on DBCloud. And to run something as complex as spike sorting, there's no more necessity to install local software. There's no maintenance for the code. Everything is already built and specification to run experiments. Some are tailored to our probes. And we want to eventually build this accessible and secure database to allow users to start collaborating. And I think an important piece of this is to build graphical tools to aid navigation across this big archive of data sets. So to automate experiments, we envision an experimenter can spend only most of their days to record data. And the next morning, when he or she returns to the lab, everything is already synchronized to cloud. And a lot of the pre-processing of the data has been done already And in this case, spike sorting is one of the most common pre-processing steps that neuroscientists do. And without this process, we've seen a large amount of data accumulated over time, over weeks or even months, without anyone having the time to do all these pre-processing steps. So behind the scene, what the cloud is doing is that it listens for new data that is written to certain directories and extracts the probe information, including channel map and impedance. And then it runs all the pre-processing steps according to pre-configured parameters. And as a new feature here, we also allow some algorithm aided by machine learning to fine-tune your pre-processed data even further. So the experimenter can quickly gain insight from this quick turnaround of data processing overnight, and this helps to aid their experiment on the next day. To set this up, it's quite simple on cloud. Really, what you need to do is to specify a local path where your data is being written to. And we see, for example, in the case of a chronic recording, each animal will have its own set of requirements, such as the type of probe that's implanted, and the parameters you would want to use for running spike sorting. So you just have to set a specific path for that animal, and then you need to choose a probe that's also associated with that animal. So in the case of using DBC probes, all of our customers will have online access to an inventory of probes that are shipped to that customer, and you can simply select from a list of probes that are already available to you. But again, I want to emphasize that this platform is agnostic to the type of electrodes that you use. So we have, vision towards supporting, for example, NeuroPixels. And once the data is on cloud, users will have this view of a list of data. You can have different folder structures to help organize your data. And by clicking on any one of them, you'll immediately have some visualization of the quality of the data that's contained inside that. So in this case, you can have some insight to how many single neurons, or the quality of the isolation of the neurons by this very flexible set of widgets. But to immediately go to here, almost through the lens of a black box, can be daunting in some cases. Most of the researcher would want to look at the raw data, and as an experienced electrophysiologist, usually they can learn something very quickly from just with a quick glance at the raw data to evaluate the signal-to-noise ratio, where the good and bad channels are, how many neurons there might be. So this interactive data viewer can be very helpful in sort of understanding the overall quality of the data. And of course, because spike sorting is already done, you can have this raster plot of the isolated units at the bottom. And also, again, all of the data is synchronized with associated hardware. So all the probe information is already built into this GUI, such as channel mapping and any impedance-related functions. And to support applications where there are a ton of channels, we see this from our customers that the number of channels are going to increase in the future. This is a unstoppable trend, and to support more than 1,000 channel view simultaneously, we have built this feature into our time series GUI to allow multiple monitors to view into the same data set. And of course, every monitor is time synchronized. And if you want to run spike sorting manually, there is a simple button you can click, and it opens up this prompt window, and it shows the list of spike sorters we currently support, which includes cluster and kilosort. And there are already a set of default parameters populated here based on our experience of what are the best options for using our probes, for the type of recording from our probes. And then, of course, the users can tweak this if they wanted to, and then just click that simple run spike sorting button, and spike sorting will run in the background. So this is in contrast to, cloning into a repository and doing all the Anaconda installations locally to support your own software. Once spike sorting is done, again, you have this flexible set of widgets to look at different characteristics of the neurons that are identified by the spike sorters. And here, as an example, you can look at the parallelograms, the waveforms, and the PCA features for the selected neurons. And you can do any of the manual curations that you would do on the local settings, such as labeling units as single unit, multi-unit, or noise. Or in this case, if you identified two neurons that should be merged, you can do so. So this is an example of a merged unit from the two units in the previous slide. And with this platform, we really want to foster collaboration across labs. You can imagine a lab that's working with a rodent model can benefit from other labs working with other species. And this platform will always be fully supported by DBC team. We have our own set of engineers on the software side and also on the neuroscience side for advisory. There are many features that are currently under development through past communications with the community. One such area is compatibility with Neuroscience Without Border formats. And also, we are more and more interested in working with clinical researchers that are working with human data and how to integrate that with the DBCloud. Then, in addition to spike-related processing, there's a lot of interest in local field potentials. So we have that under our radar also. And then more recently, we have this growing interest in applying machine learning on our platform. There are many applications that we have thought about, and today I want to bring in the second presenter, Lidor Spivak, to speak on one of the examples we have already implemented recently on automated spike sorting.
All right, fantastic. Fawn, thanks very much for that great introduction to the platform. I'd now like to welcome Lidor Spivak, as Fawn says. Thanks very much for joining us today, Lidor, and take it away whenever you're ready. Just got to unmute you there. Sorry, there we go.
Okay. Hi, everyone, and thank you, Andrew, for your introduction, and thank you, Fawn, for inviting me to speak at this webinar. I'm really happy to be here and to share my work on how to use supervised and machine learning algorithms for automated spike sorting. So my name is Lidor Spivak. I'm a PhD student at the Eran Stark Lab at Tel Aviv University. And in my research, I investigate how spike timing modify spike transmission in freely moving mice. And the main method that I use is extracellular recordings combined with optogenetic stimulation. And this kind of data, at this stage of my PhD, this kind of data is very familiar to me, but 5 years ago, when I just joined the Stark Lab, I didn't know much about extracellular recordings, and of course, I didn't know how to analyze the data. So I started by learning the pipeline of the lab, and this is a general scheme of how the pipeline looked like. So we started with the raw data, then we extracted spikes waveform from the raw data, and then we used a principal component analysis, PCA, to create a low-dimensional feature space. So this part was done with NDI Manager, which is open source software. And then the next stage was to use ClusterQuick, which is also an open source software cluster the spikes into putative neurons. So these 4 stages made sense to me, but the next stage seemed a bit strange and maybe not that scientific. And the next stage was manual curation. And in this stage, the user go over the clustering results and manually decide for each unit whether this unit is a noise unit or a neuronal unit, or whether 2 units need to be merged together, or even if 1 unit need to be split into a few subunits. So here how it looks like. So the user can look about a few units and see the mean waveform and standard deviation, and look at the cross correlation and autocorrelation, and make some decision how to curate the data. And this, of course, is very subjective to the user, and it's very time consuming. So after gaining some experience, I tried to find solution to how to overcome this part of manual curation. So I first started to look into other spike sorting algorithms. So here is a small list of sorting algorithms that are out there, but there are many more. And I didn't try all of them, but I tried few of them. And from my experience and from talking to other researchers in the field, I realized that all of these sorting algorithms still require substantial amount of manual curation. And the question is, why are these sorting algorithms still require manual curation? And I think the reason is that all of these algorithms are unsupervised algorithms, which means they are general algorithms that can work on any kinds of dataset and don't have any prior knowledge about the data. So, for example, ClusterQuick is based on expectation maximization algorithm, which is a general clustering algorithm, not just for neuroscience, for any kind of data. And it's really hard to use this kind of algorithm, which is general for any kind of dataset, and to get perfect results that will not require any manual curation, especially on complex problems such as spike sorting. So one thing that we can do is try to tweak the hyperparameters of each algorithm to fine-tune it to our kind of dataset. But this is also a really hard task. Sometimes there are many hyperparameters, and there are many combinations of hyperparameters, and it's really hard to test and to find the right parameters for a dataset. And in my opinion, that even if someone would find the optimized hyperparameters, I think the results will still require manual curation. And lastly, these algorithms are mostly based on the single spike waveform. So just to give idea how these algorithms work, so each algorithm is different from one another, but they all share a similar approach. And this approach divides the spike sorting challenge into 2 questions. The first question is, how many neurons do we have in the recordings? And the second question is, which spikes belong to which neuron? So let's say we recorded some spikes, and here is a feature space which represents this two-dimensional feature space, which represents the spikes. And each point is a spike, and we don't know anything about the data. We don't know how many neurons were recorded, and of course we cannot assign each spike to each neuron. So the basic idea is to decide randomly about a number of neurons, and then assign each spike to each neuron, and then try to optimize again the number of neurons, and so on until the algorithm reaches a conversion or a conditional stop. So it's really hard using this kind of approach to get to a perfect solution which will not require any additional step. So my idea is to use both unsupervised algorithm and supervised algorithm such that it will not require any manual curation. So So my suggestion is to start with unsupervised algorithm. And this unsupervised algorithm doesn't need to get a perfect result. Actually, it's better if the algorithm will overcluster the data. And then the next stage, we can use a supervised algorithm, which can be a deep neural network algorithm or something like that to mimic the process of the manual curation. So first we have initial clustering, which does not to be perfect. It can be very far from perfect, but then we can curate the data by training a machine learning algorithm. And this machine learning algorithm does not use the single spike anymore. It uses the initial clusters. So for each cluster, we can draw some statistics. We can draw the mean waveform, the standard deviation, the autocorrelation, and cross correlation. So we are not using anymore the single spike waveform. We're using much more information. So here is an example of the output of ClusterQuick, and we have many clusters, many initial clusters. And if the user will go over them and would label each unit, whether this unit is a neuronal unit or a noise unit, then you can use this labeling to train a classifier to mimic this process. So then in the next session, the classifier can go automatically over all of these units and decide whether each unit is a neuronal unit or a noise unit. And here's another example of a classifier that can be made. This classifier is a merge classifier. So again, we have the initial clustering from ClusterQuick, and then the user can go for few data sets and label which units need to be merged together. So here's an example of units that the user decided to merge them together. And then we can use this data, which was labeled by human, to train an algorithm to decide for each pair whether this pair should be merged together or not. So this is what I did. I trained two classifiers. One classifier was noise classifier, and the second classifier was the merge classifier. So here's an example of two units that the classifier decide not to merge them together, to keep them separate. So the classifier looks at the autocorrelation and the cross correlation between the two units and on the mean waveform and standard deviation. And for these two units, the classifier decided to keep them separate. But here are another two units which the classifier decided to merge them together. So using those two classifiers, I was able to curate a whole session, a whole data set. And here's an example of 3 units that the algorithm decided, that the automated curator decided to merge all of these 3 units together. And you can see that even though the waveform is drifting over time, the algorithm can still understand that these 3 units need to be merged together, basically using the cross correlation and autocorrelation. So I just want to show you some examples how we use it in our lab. So in our lab, we use this automated curator to make a list of units. And on the top of the list, there are the neuronal units, and on the bottom of the list, there are the noise units. And all of the units that need to be merged together are one beside another. So here we can see a unit that doesn't need any manual curation, but here are some units that the algorithm decided that they need to be merged together. So here's one example. You can see that the autocorrelation and cross correlation are very similar to one another, and the mean waveform is also similar. And here is another example. So we can see again that the mean waveform is very similar between these units, and also the autocorrelation and cross correlation. And let's look at a few examples of noise units. So here are some units which were labeled as noise. Okay. So let's look at some results. So to measure the similarity between two clustering solution, I use adjusted rand index, which is a metric that give a value between 1 and -1, and value of 1 represent identical clustering solution, that the two clustering are identical to one another, and -1 means that the two clustering are very different from one another. So first I use the raw output of ClusterQuick, and you can see that that ClusterQuick for this given session produced almost 600 units, and my clustering after manual curation, I end up with almost 140 units. And the adjusted rand index, as you can see, is very low. It's very close to 0. And then I measure the similarity between ClusterQuick and the automated curator. And you can see that the automated curator merged a lot of units. So the automated curator produced almost 160 units, and the adjusted rand index is still very low. But if we measure the similarity between the clustering of the automated curator and my clustering, you can see that the adjusted rand index is much higher. So just to give a sense about these numbers, I took 3 sessions. Each session was recorded from a different animal with a different silicon probe, and 2 of them were recorded from the C1 area and 1 from the neocortex. And I asked 4 other lab members to curate these 3 sessions. And then I measured the similarity, the adjusted rand index between all humans and the automated curator. So here you can see the similarity between humans and between humans to humans and humans to the automated curator. And on the bar graph, you can see the mean and standard deviation of the adjusted rand index for each person and for the automated curator. So as you can see, the automated curator reaches a near human level of performance, but it's not as human. So I think this is a good start. I think this have a potential to get a human level of performance. And I think there are 2 ways to improve this automated curator. So one idea is to use more data. So I think if we will have more data from and also diverse data from different species, different probes, different brain areas, so we can train an algorithm which will be very robust and could make a good sorter for any kind of data. But I think it will not be perfect for specific and unique data sets. So another solution would be to make a tailor model for each kind of data set. So if we have enough labeled data for a specific data set, let's say data which recorded from the CE1 with specific probe and were curated by specific person, so we can tailor a specific model to this kind of data set. So these are the 2 directions, and I will let Fan to elaborate more to tell us how the cloud can help us to improve the automated curator.
All right, thanks very much, Lidor, for that fantastic presentation. And now we have heard from Lidor about how AI can be used for electrophysiology data analysis. We'll hand things back to Fan, and Fan will show us how this is actually being implemented and applied on the DBCloud platform. So Fan, whenever you're ready, take it away.
Great. So as the audience have heard from Lidor about his AI algorithm, I hope you are as excited as we were almost a year ago when we first heard about this from Iran Stark Lab, showing this comparison matrix that the AI algorithm can perform near human level on curating very complex neural data sets. And this is a lab that is known for their obsession for details. So we were very excited when we saw those data. And we immediately started to work with them to see how we can implement this on our existing cloud platform to bring this to a broader audience. So here is, I'm going to show you our implementation of this algorithm. So we built a full stack feature with the backend running the algorithm silently as you as a user upload their data. Everything is spike sorted and it runs through this AI algorithm automatically. And then when the user goes back to the browser to look at this data, it is already populated with the results. So here is the view without turning on the AI results, and the typical way of looking at spike sorted results is to go through this very large list of neurons that are identified by the spike sorter. You have to go through this entire list to see if it's a neuronal cluster or noise cluster. And then for all the neuronal clusters, you have to compare against all the other neuronal clusters to see if you should merge or not. So with the AI algorithm, the user can simply click on this, toggle this switch, and this will immediately show you a set of suggestions highlighted in the list of neurons for things that the AI recommends users to curate. Here there are two different highlights. The blue highlights shows example of neurons that should be merged. And here as example for neuron number 43, the algorithm detected two other cells that should be merged. And if you're familiar with the parallelograms and waveforms, you can see that indeed these cells should be merged. And this is the case where the AI actually identified two clusters that should be merged, but these clusters actually look quite differently on the waveform view because this is likely coming from a bursting pyramidal cell in the CA1 layer, which is a very often recorded region by the Stark lab. So the algorithm has learned to mimic the human decisions to merge these two seemingly different clusters. This is what we have also observed as a common mistake done by unsupervised spike sorters nowadays, to mistakenly split these into two different clusters. And here is example of a cell as labeled in gray, and you can tell from the parallelogram and waveform that this is indeed a noise cluster that you should throw away without further analysis. And I think as Lidor had hinted that the AI algorithm we have implemented on cloud so far was trained by user input from his lab, which is very specific in the sense that it's almost exclusively coming from mouse and from either neocortex or CA1 layer in the hippocampus, and using silicon probes from DBC. And they have their own sorting techniques. So we have implemented this new way of allowing this entire workflow to allow users to train their own specific and tailored AI models for their own research techniques. So from the front end UI, from the user interface, the user simply needs to curate their own data sets. And once every single neuron is labeled and the ones that should be merged are merged, users can approve this curation session. And this then goes into a list of approved sessions, which if you have at least 5 of them, you can use those sessions to train a new model that you can use for all of your future research. So, and also, we think this is a bigger challenge, but if we get enough number of data sets and also from a diverse set of species, it is our vision to one day generate a universal spike sorter that can handle all sorts of data sets. So to summarize the presentation today, we have already built the infrastructure on cloud to perform very fundamental tasks such as storage, analysis, and sharing of data, all on cloud, and you can have access to all of this through a browser. And we have our eyes keen on integrating AI features. There are a number of things we are actively thinking of implementing at the moment. And just to give you some examples of custom applications that we're very interested in working on, the first one is how can we combine large channel count, high density electrophysiology data, combining with AI algorithm for drug discovery. So this will be working with preclinical animal models for early stage drug screening by detecting electrophysiology biomarkers. And in the second case, we are also working with collaborators on clinical applications where, for example, in the epilepsy monitoring unit, where both the hardware and software technologies are still far behind the current academia, standards using these FDA-approved protocols. So we're looking at ways to improve that in the future also. So if you are currently working with any of these applications and you can see how you can benefit from either the existing DBCloud platform or extension of our features, please reach out. We encourage you to look at this handouts tab that should be present on your webinar interface. And from the handout, there's a link to our website where you can schedule a meeting for follow-up and learn how to sign up and subscribe to our service. Or you can just directly email us at info@diagnosticbiochips.com. Thank you for your attention today. I'll let Andrew wrap up the webinar.
All right, thanks very much, Fawn and Lidor, for those fantastic presentations. We're actually now going to move on to the Q&A portion of the webinar, and it's now my pleasure to welcome Fawn and Lidor back. And thanks very much to both of you for those great presentations. And if you're ready, we'll jump into some Q&A.
Yeah.
All right. So here's a great question to start things off, and you touched on it there towards the end, Fawn. But I think I know what the answer is, but can DBCloud support analysis using high channel count MEA data? I'm guessing multi-electrode array data. Yeah.
Right. It's actually a very common question that we get asked all the time. Yes, the short answer is yes. Our platform is agnostic to the type of electrode that you want to use. And this extends beyond MEA to other type of in vivo electrodes. I mentioned NeuroPixel, but any of our competitor probes with the right data type format, we can support running on cloud.
Okay, fantastic. Another great question here. I think this one is more for you, Lidor, but what are the features? You mentioned some features that the AI is using for classification. What are the features that the AI classifier uses for training and predicting? And again, similar, is it agnostic to the type of spike sorter used?
Okay, so first of all, the features are based on the mean waveform and standard deviation. And also the autocorrelogram and cross-correlogram between units, or for the noise classifier, it's just the autocorrelogram. And this is like the simple answer, but there are a little bit more tweaking for the features, but I don't think it's necessary to detail all of them right now. And for the second question, this is something that we are not sure yet. So right now, the algorithm was trained on a specific data set, which we train on just ClusterQuick. But I think that if we would have enough data set from Kilosort or any other kind of algorithms, we can train a new model that I believe that will work better and will be agnostic to any kind of sorting algorithm.
Okay, fantastic. This is a great question. So despite the recent growth of automatic spike sorting tools, many of which you've discussed here today, a great proportion of researchers still do it manually. Why do you think that's the case?
That's a great question. Let me start on this, and maybe Lidor will fill in from the perspective of actual neuroscientist. So from our point of view, we interact with a very diverse, set of end users, and each user does spike sorting a little bit differently. Their recording has different challenges, different animal species, different noise level. So there's all sorts of different pre-processing done differently to your data sets. And also, I think a big challenge in spike sorting is that there's almost no ground truth. So it's very hard to evaluate the quality of spike sorting. So then if someone proposes a general universal approach, it is very hard to then validate and implement it for your own use. So in our view, DBCloud is can be a great solution to tackle this very challenging problem. So you can, as Lidor said, you can either train your own AI algorithm to tailor, towards your own specific setup, and the goal is to have the AI perform as good as your own experimenter, so that you can rely on this automated process to save your curation time. And the other is to create this universal one solution for all AI model for everything. But maybe Lidor will speak on how realistic that is. And yep.
Yeah, so I think this is like this is the answer. Like, if I think most of the sorting when they are designed, most of the spike sorting algorithm when they are designed to be universal, to sort all kinds of data sets. And I think this is like really hard. And also, as Fan mentioned, there is no ground truth. So usually using synthetic data to evaluate the performance. And the idea is to try to make, and the idea of the other sorter is to make a sorter which will work as good as the ground truth. And we're proposing something a bit different. We are proposing something to mimic the curation process of the person. So it's something, if the person is not experienced and the algorithm will train an unexperienced person, so the curation will mimic this person, but probably this solution will not be that good. But on the other hand, if we have someone experienced, which labeled the data, and we can use this high experience labeled data, we can train a really good algorithm to mimic this process. So I think this is two different approaches. One is to make a universal sorter, which will sort any kind of data set, and my approach is to mimic the manual curation. And I think if we have enough data, this is a question that I'm not sure about it, but as things looks like now, that advancement also in AI, if we have enough data and we can use a really good an AI model, so maybe we can make a model that will be able to mimic, to be able to handle all kinds of data sets.
Okay, excellent. Great answers. A great question as well. Thank you for that. This is, I think, a simple sort of methodological question. Can the automated curator split units in addition to merging units?
Okay, so in principle, everything that the human can do, so we can try to make an algorithm to do so. Right now, this implementation, the algorithm is not able to split units, only to merge or decide whether a unit is a noise unit or a neuronal unit. Just for simplicity, because those two things are a binary decision, it's a binary classification, whether it's a noise or neuronal, or whether to merge or not merge. But I think we can try also to work on splitting units, which is not a binary. You need to decide how to split or where to split them. So it's something which is a bit more complicated, but it's doable if we have enough data, which labeled by a human, so we can train an algorithm on this data, and I think we can also achieve that.
Okay, excellent. There's a question here about the types of data files that can be analyzed using DBCloud. For example, can you upload neuroshared data?
Yeah, I think this is similar to the first question regarding how compatible our platform is across different electrode type and data format. So as it stands, we currently are agnostic to electrode, but the data format is supported is the very flat binary format used by Intel and OpenEFIS. But if you have any other data formats, we can certainly discuss and support it. It's not a technical barrier to do that. We just don't want to host, too much options on our platform. So one of the goals here is to standardize EFIS pipeline. So there's a trade-off between how many format we support versus standardization.
Okay, yeah, great. That's a great point. This is a great question here in terms of specific applications. Can the spike sorter AI keep track of what happens to a specific cluster of spikes before and after a treatment, for example? So, could it be done by linking amplitude and timing? And basically, if the researcher wanted to answer the question of whether a cell has decreased or increased their firing rate due to treatment. Is that something that's doable?
Yeah, I think it's doable, but I'm not. This is a okay. So it took me time to understand the question. I think it's the question is, if I understand correctly, it's not related to the AI sorter that I presented. Maybe it's a different application of AI. And I think in general the question is, if there is if human doing it manually, so I think we can train a model to do this kind of task. So right now at the stage, like AI is really advanced and really hard stuff, like I don't know, maybe like driving or writing complex text, but even this kind of stuff AI can do. So I think this kind of linking firing rate or amplitude over time, if there is data which was labeled by a human, so we can train an algorithm to do that as well.
And I guess the answer too is if anyone has a specific application in mind, reach out to you and you're happy to discuss it as well, right? And
I just want to add to that. Like, let's say you have some experiments where you typically see cells that drift. And maybe through manual curation, you have a technique to identify this drift and you would want to merge them. And perhaps the AI can pick up on your technique in identifying these drifting cells. So maybe without specifically tracing, the amplitude change or timing or of the cell, the AI can give you the solution, which is to actually decide whether these clusters belong to one cell or multiple.
Yeah, so yeah, definitely in our lab we use optogenetic stimulation and usually to increase firing rate in cells. And yeah, the algorithm works really good on merging units, even though there is a changing in firing due to optogenetic stimulation or any kind of behavioral change or something like that.
Okay, perfect. A quick question here: can we get the software as a standalone product?
Yes, definitely. There are several options for subscribing to the service. You can certainly get the software as a standalone product. For example, if you use other type of electrode, or we offer this hardware software package for any DBC related probe. So please contact our salesperson using the link provided under handouts, and we will reach out to you with a more specific answer.
Okay, great. There's a great question here about, can you talk about how you would prevent overfitting? Is there a danger of that?
Yeah, there is danger of that, and I think the answer is a huge amount of data. Yeah, there is a danger of overfitting, and I think in our lab we had manual curation for a few years, so we had a huge amount of data, and this amount of data I think help us to prevent overfitting. And yeah, I hope that with the DBCloud we will be able to get much more data and work on overfitting.
Okay, excellent. This is a really nice question here. Someone writes in that they've got interest. The challenge is finding software that can help them overcome their lack of expertise, and that they've done many MEA recordings, but they stopped because they didn't have confidence in their ability to sort the spikes accurately. And it can be a challenge to find outside experts or time of those experts to train in the spike sorting. So if for groups that maybe don't have their own personal knowledge or expertise with the manual spike sorting, can the platform fill that gap?
Yeah, I'm glad you asked. This is the exact problem we're aiming to solve, especially towards less experienced users, and also with MEA, there's a ton of channels. So I imagine spike sorting is a daunting task. So we do provide, the platform as it is, it's pretty easy to use. So I encourage users to experiment with it, but we do provide support, one-on-one support to walk you through your research. And if you have any need, either through cloud or, we can provide all sorts of technical support.
Okay.
So I want to add just one more thing. As I mentioned, in our lab, we use the automated curation as a recommendation, to give us recommendation. And new students that came to the lab also learned from that a lot. So there's nothing that can replace an expert that sitting beside you and explain everything. But even with an expert, using this recommendation can help someone with no experience to understand the logic. So the algorithm gives some recommendation, and then the user can understand based on the recommendation what is the logic behind the sorting process. So I think the combination of using a bit advice from experts, but then using the recommendation could be a great combination.
Okay, fantastic. Almost out of time, but I think we can squeeze in a couple more questions quickly. One is, can the sorter handle multi-session data and identify units that appear on several recordings?
Sorry, can you repeat the question?
Sure. Can the sorter handle multi-session data and identify units that appear on several recordings, different recordings?
Yeah, I will chime in here, but Lidor should maybe speak from his experience. But in our case, some customers are okay with merging different sessions into a single file and run spike sorting that way. But in other cases where I think different sessions are always treated as independent data sets, you cannot guarantee one unit found yesterday can be confidently associated with the same neuron found today. So there are two different data sets completely isolated.
Yeah, I agree with Fan. In our lab, we use the second approach. We treat each session as a different session, each day as a different session. And yeah, the algorithm right now don't track units over different days.
Okay. I think we have time just for one last question. Fawn, you mentioned about the application of the probe and software technologies to preclinical drug screening. Can you just talk a bit about what that plan actually looks like?
Yeah, so we have talked to many pharma and CROs, and we learned that there's a huge need in preclinical drug screening process because there's a very high failure rates going from that to clinical trials. And if you can minimize that failure rate, you can save a ton of money and time in drug development pipeline. And I think one of the issue with the current preclinical model is that, first of all, your model needs to be validated, but also right now, the typical approach is either doing single cell analysis with things like patch clamping, or on a macro scale, like using behavioral models to look at something that's maybe not directly related to the underlying disease mechanism. So we believe it's important to use electrophysiology to probe the specific neural circuit that's underlying or is correlated with disease. So if we can identify such electrophysiology biomarker in that specific circuit, then we can look at animals in healthy, diseased, and states where drug is dosed. So that's our vision, and also combining with AI, I think we may be able to quickly detect these biomarkers, either known or novel. So if you're interested in working along these lines, I highly encourage you to reach out to us, and we can see how we can develop a project together.
Okay, amazing. So perhaps more opportunities to scale up and speed things up, I guess, in particular with the AI. Wonderful. Unfortunately, we're out of time. I'd really like to thank both of you for your time and sharing your insights with us today. It's been an absolute pleasure having you with us.
Thank you, Andrew.
Thanks very much, both. And of course, a big thank you to everyone in our audience as well for your participation today. Last but not least, we'd like to thank Diagnostic Biochips for making this event possible. In closing, we hope you enjoyed this Inside Scientific and Scientist.com webinar, and we'll see you again next time. Thanks, everyone.